Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening. I'm Yvonne Stapp for Science for the Public, and I welcome you to this evening's program, Contemporary Science Issues and Innovations. Tonight, we're honored to have as our guest Sheldon Krimsky. Sheldon Krimsky is a professor of urban and environmental policy and planning in the School of Arts and Sciences, Tufts University, and an adjunct professor in public health and family medicine at the Tufts School of Medicine. He also teaches at the New School in New York City. Dr. Krimsky's list of accomplishments is so lengthy that I must limit this introduction to only a few highlights and refer you to the Science for the Public website to view his many distinctions. He brings to his field a background in both science and philosophy, and that combination may account for his unique perceptiveness. For decades, Dr. Krimsky has focused on the ways in which scientific advances often create unanticipated health concerns and ethical issues. For example, he alerted the public and policymakers early on about the health risks of endocrine disruptors such as BPA. He warned about the potential medical and ethical risks of biotechnology, especially in genetic splicing and GMOs. And he's been a leading voice in alerting the public about how the DNA databases can violate very basic rights. He's a founding member and board chair of the Council for Responsible Genetics. When considering Dr. Krimsky's work, it's good to recall how serious warnings have so often been ignored or ridiculed, both by vested interests and a complacent public. We're easily reminded of how vested interests undermine those who warned about tobacco, toxins such as DDT, PCBs and BPA, and nuclear power plants, ozone depletion, genetically modified foods, and climate change. In all his endeavors, Dr. Krimsky's work represents a major contribution to the improvement of public policy, and his work underscores why citizens today must make the effort to be well informed. Tonight, Dr. Krimsky focuses on DNA databases. We're inclined to think of the great benefits of DNA data, for example, in medicine, family planning, and identification. However, there's a darker side to DNA databases, and Dr. Krimsky's recent book, Genetic Justice, co-authored with Tanya Simoncelli, details how such data can be and is misused. An important feature of this book is the comparison between DNA policy across several advanced nations, which makes the reader aware of the possible shortcomings of American policy. The book is an excellent example of the importance of public understanding of scientific advances that may initially seem benevolent but can raise serious ethical issues. As complex as the subject may seem, it's an essential one to know about today. It is presented in this book in a clear and interesting manner, and readers will be grateful for such a resource. It's a great honor to welcome Dr. Krimsky, not only as an outstanding example of academic research in the public interest, but as a hero to the advancement of science literacy in this country.
The title of this presentation is, What's Your DNA Profile Doing on a Federal Database? And I'd like to start off with a few questions that you may not really have considered. First of all, did you know that in some states police can obtain an involuntary sample of your DNA of an individual who has not been charged, convicted of a crime, and upload his or her forensic DNA profile on a state or national database. In England, this happens to children as young as 10 years old. Did you know that police can follow you around, surreptitiously collect an object that you discarded, obtain a cell residue from the object, develop your forensic DNA profile, upload it onto a local database, all without a warrant or your permission? Did you know that when police collect voluntary DNA samples from a large number of people in a community, let's say to exclude them from being a suspect in a serious crime, that's called a DNA dragnet, the forensic DNA profile is kept permanently on a police databank without permission of the innocent volunteer, even if the person is completely not implicated in any crime. Did you know that if police find a close but not an exact match between crime scene DNA and a forensic profile on a DNA database, they can investigate the DNA of an entire family without a warrant or any individualized suspicion? Did you know that there have been over 250 individuals who have been exculpated of their felony convictions and released from prison on the basis of new DNA evidence? In one case, after a person was executed for a crime, it was determined that his DNA did not match the crime scene DNA evidence. That's the first case that we know of where a person was executed by the state who may not have been guilty of the crime. So the key questions for ethics and civil libertarians is, to what extent and in what context do and should people have a right to claim privacy over their DNA? And how do DNA privacy considerations compare in medicine versus forensic? Some of you may not know, but there's a lot of protections for your DNA in medical science, but very few protections when the DNA is obtained by the police. What role, if any, does the changing technology play on questions of privacy? And how do we balance the roles of personal privacy and societal considerations when we're dealing with medicine versus forensic DNA? These are some of the things I would like to discuss today. But first, I want to help you understand what a forensic DNA profile is. And hopefully I can take you from the very rudiments of this idea to how it is done by the police and their laboratories. Your forensic DNA profile consists of 13 pairs of numbers obtained by analyzing the DNA in your chromosomes. So each of us, theoretically, has a profile that consists of 13 pairs of numbers in the United States. It might be slightly different in other countries. So how do scientists and technicians obtain the forensic DNA profile, or how do they obtain these 13 pairs of numbers? It starts with taking a biological sample. That could be blood, semen, hair, saliva, teeth, bone, any tissue in your body. As a matter of fact, if I take my thumb and press it on this table and then leave it, a forensic scientist can come by and swab it and probably get enough cells to be able to obtain a forensic DNA profile of myself. So that's the starting point, a biological sample. The sample is then taken to a forensic laboratory where technicians separate the DNA by chemical and physical processes. The DNA is extracted from the cells so that it is pure DNA. It's then put into a forensic DNA analyzer. It's a huge, big black box which selects certain pre-assigned segments of our chromosome at 13 sites and measures the number of repeated chemical segments in those sites. So this is what the DNA analyzer looks like. It's not a very large piece of apparatus, and this is found in every school of criminal justice where forensic DNA science is taught. Now, each of us has 23 chromosomes. Actually, we have 46 chromosomes. We get 23 from a father and 23 from our mother. And what you're seeing is 23 of those chromosomes on the screen. And the criminal justice system in the United States has decided that they would find 13 sites on these 23 chromosomes. And these would be the sites that they would look at to determine the profile, the forensic profile of an individual. So you'll notice, for example, in chromosome 2, there's a site called TPOX, right here. That's a code name for forensic scientists who know that they can excise the information in that site and determine what the numbers are for that particular site that make up your 13 pairs of numbers. All DNA is made up of 4 basic chemical units, abbreviated as A, G, C, and T. Therefore, any DNA sequence in our cells is comprised of some combination of these 4 letters. And at a particular site, let's say going back to the one I just showed you, this particular site, they excise all the chemicals and they count the number of repeats at that particular site. So person 1 might have 3 repeats of AGCT at that site, whereas person 2, somebody else, might have 5 repeats. So how does DNA differ among individuals? Well, the forensic DNA profile differs by the number of those chemical repeats. So I've given you 3 persons and on this chart, and the particular repeated DNA is AGCT, and the numbers under it indicate how many times it's repeated. So you can see for person 1, there are 6 repeats of AGCT. Person 2 has only 5 repeats, and person 3 has 7 repeats. So we're talking about taking a piece of your chromosome at a particular site and figuring out how many repeats in that particular site of those 4 units, A, G, C, and T. What does your DNA profile look like? Well, because you receive one chromosome from your mother and another from your father, at any chromosome site you will have 2 numbers. One number representing the repeats given to you by your father's chromosome, and another number representing the chemical repeats from your mother's chromosome. So I've placed one particular series of numbers, 13 pairs, which represents the repeats and therefore the forensic profile of an individual. 7'5", 6'3", 4'7", etc., etc. Each of us, if we had our DNA analyzed, would have 13 pairs of numbers that would be different. That's our forensic DNA profile. What our DNA can reveal in general, once it's understood, we are now beginning to realize that our DNA can reveal certain things about our disease states, whether we have an inherited genetic disorder, or whether we have certain predispositions toward diseases, mutations that correlate with the onset of a disease.